Hi folks, welcome to chapter 3 of our video series where we are analyzing head anatomy for an anthro and figuring out how to make a transformation work within our lifetimes. In chapter 1, we described what an anthro's head would look like and how things like speech, facial expressions, physiology, and overall shape would intersect with anatomy. When needed, we will briefly review relevant aspects from chapter 1 here, but we would still recommend that you watch it at some point if you haven't seen it yet. In chapters 2 and 3, we are going into depth with anatomical and tissue changes that are needed to go from an adult human shape to an anthro's, and connecting those to relevant techniques that can be used. These videos aim to help you understand what's going on at the level of biology and develop an intuition of how changes will work in that context. In several situations, we make proposals for how changes can be performed. Some of them have higher confidence, while others are more speculative. These represent our current thinking for the most promising directions of research to explore, develop technology, and bring into reality. We have been dividing our discussion across several tissue systems. Bones, cartilage, and muscles and tendons were in chapter 2, and the nervous system and the visible portion of the eye, that is the anterior segment, are in this chapter 3. Again, we're not covering some other tissue systems. We won't get to adipose tissue, secretory glands, respiratory membranes, or teeth. Also, we will not be discussing skin, fur, scales, feathers, whiskers, and so on, since those are being handled in our integument review project. The nervous system lets animals collect, process, and act on information from the environment, both externally and internally. Yeah, that's obvious, but it's still important, and the nervous system is absolutely everywhere in the body. The brain and spinal cord are where a lot of that information processing happens. As we established in chapter 1, we're going to stay away from changing the shapes of the brain and spinal cord for our wolf anthro here, so we are going to treat those organs as black boxes. Information is gathered by sensory organs, whereas actions taken by the nervous system are implemented using motor functions, such as muscle movement. Nerves are what animals use to transmit information from one place in the body to another, specifically and fast. Nerves are extensions of the overall nervous system and connect the brain and spinal cord to basically everything else in the body in specific ways. If information is getting collected by the nervous system, it's transmitted inwards by sensory fibers, whereas if actions are getting taken, those instructions are transmitted outwards by motor neuron fibers. In terms of how we organize this discussion, you know the drill by now. First, we will name specific tissues involved. Next, we will cover some relevant biology to help understand the system, and finally, we will propose ways of working with this tissue. Following our pattern of analysis so far, let's discuss regions in the craniofacial nervous system and see which ones are relevant to a transformation procedure. There's 3 key places where the nervous system may have to adapt in some way for a changing body: within or around sensory organs, within or around the targets of motor neurons like muscles, and along the paths of nerves. The head has a lot of sensory organs in it, but we only need to think about a few of these organs with respect to an anthropomorphic transformation. While in no cases are we aiming to increase the sensitivity of these senses for our first anthros, just to keep things simpler for now, we do need to be aware of any changes to the shapes of these tissues. Crudely, there's sight, sound, taste, smell, and touch, plus the senses of balance and proprioception. In the chapter 1 video, we established that the inner ear does not need to shift or move. The sensory organs there, sound as well as your sense of balance, are therefore all set. Also in that video, we established that we're probably going to stay away from modifying tissues responsible for the senses of smell and sight. That means that the relevant organs, the olfactory epithelium and your retina, should stay roughly as they are without tissue shape change. This leaves taste, touch, proprioception, and some general sensory feedback from the odd organ here and there. There's too many tangential details to go into at this time. All of these organs have distinct and very specialized structures. So if you're curious, you can check out things like Pacinian corpuscles, Meissner's corpuscles, Ruffini's corpuscles, free nerve endings, and muscle spindles to see how they work. And you can probably guess that we want to keep the existing ones intact so that we don't have to make more of them. That should be doable since these organs are all self-contained. Their surrounding tissues will adapt without requiring manipulations to these organs. Then for motor organs, we're mainly talking about muscles like skeletal muscles and smooth muscles. There's some edge cases here, like some secretory organs, but even for things like salivary glands, they are controlled in some way by smooth muscle activity. In any case, the motor tissues that will experience shape change are mainly limited to muscles, skeletal muscles, and smooth muscles. We already discussed shape change of the skeletal muscles themselves in Chapter 2, so insofar as the nervous system is concerned, we're just thinking about the NMJ, the neuromuscular junction. And you only need one NMJ per muscle fiber. For the existing muscles in the face, they already have all the NMJs they need. We don't need anymore. Therefore, we do not have to add skeletal muscle target tissues to our list for the nervous system. Then for smooth muscle cells, they get pretty dense motor neuron innervation. However, as we established in Chapter 2, there's not that many smooth muscles we actually need to worry about since most of the relevant tissues, such as vasculature and associated smooth muscle constrictors, are capable of remodeling their networks to extend fully functional new tissue to changes in physiological needs. The only smooth muscles we need to think about are the ones that control gland secretions as well as iris constriction. For the sake of this nervous system discussion, we will aim to keep constant the motor neuron junctions on these organs and not plan on adding any new smooth muscle units. So that covers the sensory and motor tissues. We need to be attentive to tissues that have sensory organs providing touch, proprioception, and taste, and also be careful to not mess up sites of motor innervation. But in all cases, these structures are broadly distributed, self-contained organs throughout highly adaptable tissues. Changes to the surrounding tissues, if done right, will have negligible impact on these nervous system structures. Now for the nerves themselves. Let's start with the nerves that will not be impacted by a changing body to get them off the table. Cranial nerve 0, the terminal nerve, and cranial nerve 1, the olfactory nerve, mostly stay inside the brain case, and as we've previously established, we're not changing the shape of the brain case. Cranial nerve 8, the vestibulocochlear nerve, receives signals for sound, balance, and head movement, and it never leaves the brain case either. As we learned in the first video, the inner ear, which this nerve innervates, does not need to move. As well, the accessory nerve, or cranial nerve 11, stays inside the neck and is ultimately responsible for stimulating some muscles in the neck and upper back. Similar to how we're avoiding modifying the brain case, we're not going to substantially modify any deep tissues in the head or neck through which CN11 passes, so CN11 is off the table. Going down from the cranial nerves, we start to get into the spinal nerves. Spinal nerves occur at regular intervals as you go down the neck. While we can't take the first 3 cervical nerves off the table, we can mostly ignore everything starting with the 4th cervical nerve and afterwards. Next up on the triage, we've got some nerves that will be impacted in part by a transformation, but not to a large degree. Maybe these nerves need to become just a little bit longer, or maybe they're in regions that a surgeon would need to work around due to some other procedure being done. Basically, these just need to be in the backs of our minds for now. These include the nerves that supply muscles that move the eyes, the oculomotor nerve, trochlear nerve, and abducens. While the eyes will need to be advanced forwards, these nerves have fairly simple paths through the soft tissues inside the orbits, and due to their peripheral nature, they should adapt to shape changes faster than the optic nerves, i.e. the oculomotor, trochlear, and abducens nerves will probably not be rate limiting, and therefore not be vulnerable to damage as their anatomical context changes shape. This will make more sense when we get into the biology of nerves shortly. Next up, there's the glossopharyngeal nerve. This nerve carries some sensory and motor functions for muscles in the mouth and pharynx, and carries sensory information from part of the tongue. The tongue will need to be longer, but this is not exactly the most complex cranial nerve either, so this nerve gets a maybe. Another maybe comes from the vagus. The vagus is deep inside the neck and mostly does things we're not interested in here, but one of its branches innervates a muscle at the back of the tongue, the polyglossal muscle. The rest of the vagus, including its main trunk, is not impacted. Rounding out the low priority nerves, we come to 3 of the cervical spinal nerves. The first cervical nerve supplies motor information to the geniohyoid muscle on the floor of the mouth, and this muscle will need to be longer. And portions of the second and third cervical nerves accept sensory information from the skin of the scalp, cheeks, and under the ears. The scalp isn't likely to change in size much, but the cheeks might. Even so, all of these nerves are relatively simple in their functions and target tissues, are peripheral in nature, and therefore easier to work with, and we'll see relatively smaller changes compared to the nerves that we're going to talk about next. We've taken some things off the table, but we are still left with 4 nerves that need attention. The optic nerve, or CN2, trigeminal nerve, CN5, facial nerve, CN7, and hypoglossal nerve, CN12. To be sure, the optic, trigeminal, and facial nerves are very large and complex, so being able to take other nerves off the table doesn't mean we're getting away scot-free. The optic nerve will need to be longer due to anteriorly moved eyes and orbits. The trigeminal and facial nerves branches will need to be much longer due to a longer snout, and the hypoglossal nerve will need to be longer due to a longer tongue. But don't let the large branching structures scare you either, because we've got some tricks up our sleeves. So let's discuss the biology of these nerves a bit more and figure out how amenable these tissues would be to large-scale shape change. Nerves group together a lot, thousands of axons, using a hierarchical structure. Starting from the outside, there's various layers of connective tissue, then you see several fascicles. Each fascicle contains its own bundle of axons, say a few dozen or 100. And finally, within these fascicles, axons are organized in 2 main ways. Some axons need to transmit information really fast and get an insulating sheath called myelin. You've probably heard of Schwann cells at some point. They are responsible for making this myelin sheath in the peripheral nervous system. Myelin sheaths in the central nervous system, including the optic nerve, are similar, but are made from a different kind of cell, oligodendrocytes. Other axons transmit at a lower speed and are not myelinated. Instead, those axons are grouped together by Schwann cells in the peripheral nervous system that is a different kind of Schwann cell that is not myelinating. Zooming in a bit more, let's look at the cells and molecules making up these structures. The outermost nerve sheath, the epineurium, is mainly built from collagens type 1 and 3 and is home to fibroblasts, helping maintain the extracellular matrix, as well as some adipose cells to help cushion the nerve. As well, blood vessels, lymph, and sensory nerve fibers course throughout the epineurium. Deeper in, there's the perineurium, composed of many layers of perineurial cells and extracellular matrix, a mixture of collagens, including collagen type 4, fibronectin, and laminin. The perineurium is an important diffusion barrier. As well, the supply of blood vessels, lymph, and sensory neurons continues into the perineurium. The perineurium defines the boundary between different fascicles within a nerve. Finally, there's the endoneurium, which forms boundaries between axons within a given fascicle. The endoneurium's matrix contains collagens that are mainly of the type 3, 4, and 1 variety, and a few other structural molecules. At least some of these collagen fibers are oriented parallel to the nerve's long axis, by the way. The major cell type in Endomyrium is Schwann cells, so in addition to encapsulating axons, the Schwann cells help build their surrounding matrix. Additional components include fibroblasts, blood vessel cells, a few immune cells, and the terminations of sensory neuron fibers monitoring the nerve's tissues. Reaching the level of myelinating Schwann cells, these cells have many concentric layers of cell membrane attached to each other and to the axon held within. Getting close to the nodes of Ranvier, these concentric layers angle inwards with each of their edges attaching to the cell's membrane. Finally, at the nodes of Ranvier, the outermost layers of the Schwann cells terminate, leaving a tiny portion of the axon's membrane exposed to the outside. But the connections between neighboring Schwann cells are tight, so the exposed region is quite small indeed. Nodes of Ranvier are very specialized. Ion channels are highly enriched at nodes of Ranvier, and this is where saltatory conduction gets made possible. We will skip the details of how the voltages in ion channels work, but what we cannot escape is the fact that the structural connections between Schwann cells and axons at nodes of Ranvier are very specialized and are tightly coupled to the ability of axons to transmit information. Ultimately, we would rather not touch nodes of Ranvier nor engineer new ones if we can avoid it. An important detail is how far apart these nodes of Ranvier are. There's this idea of a saltatory conduction safety factor. The internode distances are shorter than what they need to be, providing robustness in excess of what is needed for successful transmission of a signal along a myelinated axon. The safety factor is generally 5-fold or greater in healthy myelinated axons. So we expect that you can extend the nerves we've implicated up to a limit imposed by the internode distance at which conduction is no longer reliable for myelinated axons. Crudely, this limit could be, for example, around 2-fold lengthening based on a study in adult rats. More testing should be done that explores the optimization of nerve extension rates and that checks whether various interventions fully preserve nerve conduction. In contrast, by definition, for the axons that are not myelinated, nodes of Ranvier are not a concern. Finally, in terms of the axons themselves, there are several structural molecules that make them up. These include actin, microtubules, and spectrins. These molecules are made of polymers far shorter than the axon's length that connect with each other in networks that collectively extend the axon's whole length, which is to say we do not need to worry about specifically preserving the connectivity of any particular polymers inside the axons. To distill down the biology of nerves, we have connective tissues, some supportive tissues like blood vessels, and neurons and glia. The connective and supportive tissues are expected to easily remodel and adapt to changes in shape and size of their surrounding anatomy. Meanwhile, while we want to avoid modifying nodes of Ranvier, we can extend internode distances by quite a bit. Going into the molecular level, this is tractable because the vast majority of axons' lengths are composed of fairly uniform and networked molecules. There are no contiguous polymers that are critical to preserve. In the future, methods that can add new Schwann cells and increase the number of nodes of Ranvier would also help, of course. We just don't want to rely on them for our first anthros. There is ample qualitative data on peripheral nerves being able to extend and adjust in response to a source of gradual external strain. Nerve injuries are lowest when tissue expansion occurs at a slow rate, such as less than 1 millimeter per day in the human mandible. The axons and Schwann cells of peripheral nerves are capable of remodeling and extending, growing alongside the surrounding tissue as long as it's done carefully and they're given the time they need. While direct quantitative data is limited, the internode lengths for several cranial nerves are generally less than 1 millimeter, and we know from a different setting in adult rabbits that internodes can be lengthened by at least 22% from their baseline, reaching nearly 1.5 millimeters in rabbits while preserving nerve function and axonal diameter. And again, there was that 2-fold number from rats we mentioned earlier. As a consequence, we can safely extend craniofacial nerves beyond their current length, while keeping the number of nodes of Ranvier the same. This makes our lives much easier. No new Schwann cell engineering needed. We expect the only real limit to extension will be that of maintaining reliable saltatory conduction, since the tissue appears to remodel adequately otherwise. This upper limit should be explored more, of course. But how about the optic nerves? Data is more limited. In molecular terms, axons and optic nerves are similar to peripheral nerves, including actin, microtubules, and spectrins, for example. We assume as long as the cells are given enough time to remodel and adjust to increasing lengths, that the upper limit to extension would be defined by maintaining reliable saltatory conduction. There's at least one paper that seems promising. Yeah, it's a thin field currently, but ultimately this is something we will have to investigate more and find out if any interventions are needed to, for example, cause retinal ganglion cells to increase production of proteins needed for remodeling during axon lengthening. Overall, the peripheral nerves, the trigeminal, facial, and hypoglossal nerves are expected to be fully capable of extending over time to adapt to a longer snout and other relevant shape changes, potentially up to around twofold without affecting conduction and without requiring more advanced cell manipulation techniques. The optic nerves might be similar, but less is known about their response to gradual stretch compared to peripheral nerves. A boost using genetic vectors or growth factors may be useful to get the axons and myelin sheaths to extend faster than they would otherwise. So overall, what do we need to keep in mind about the nervous system? First, there are many sensory and motor organs in the head. We want to keep these intact and avoid disrupting connectivity as much as possible. Some changes in their size or shape should happen automatically as needed, though. More prominently, a few key nerves in the head and neck will need to extend, especially the optic nerve, trigeminal nerve, facial nerve, and hypoglossal nerve. In terms of feasibility, axons within these nerves should be able to be extended as their cytoskeletal molecules gradually turn over and get replaced with longer polymers. This happens naturally even without providing external growth factors. And finally, at some point, a limit to axon extension will be the distance between nodes of Ranvier, at least until ways of generating new nodes of Ranvier are found. And finally, we come to the eyes. As a reminder, in Chapter 1's video, and at least for an Anthro wolf, we said that the pupil's size and shape probably do not need to change for proper aesthetics. Also, we had said that an iris might not need to be larger, potentially helpful, but perhaps not critical, depending on how the surrounding eyelids combine with the overall visual impact of the eyes. Of course, being able to enlarge the iris and pupil, as well as changing the shapes of pupils, may be beneficial for anthros, especially other species besides wolves, so this discussion is certainly worthwhile to have. We made some other smaller comments as well, including comments on the tear glands around the eyelids. Due to complex connectivity within the existing tissue, we think that eyelid modifications for an anthro, like giving them a little bit of fur, need to be performed in situ rather than swapping out entire eyelids via tissue grafts. This is honestly like a lot of tissue systems across the body, where a lot of the times, though certainly not all, it makes the most sense to modify in place. So aside from surgeons working in the area needing to be careful about not disrupting the glands, we probably don't need to spend much further thought on the eyelids and surrounding tissue. Therefore, here, we're focusing on the anterior segment of the eye so that we can figure out what tissue changes pertaining to the iris and pupil might involve. More specifically, we will narrow in on the sclera, cornea, iris, and pupil. All of these are necessary to discuss as part of considering the appearance of the iris and pupil. The iris is most interesting, but we really have to start with the sclera first. The sclera forms the whites of the eyes. It provides most of the structural strength of the eyeball and constitutes most of the eyeball's surface. The sclera is mostly made of dense layers of tightly packed collagen and other structural molecules that are laid down by fibroblasts. Most of that action takes place during embryonic development, and there are relatively few live cells within adult scleras. Overall, we've got a very strong tissue that is mostly composed of non-living structural molecules. Similar to some other tissues, that means that we cannot expect the sclera's shape to adapt if we were to apply forces to it. The tissue will not remodel. If you need a larger iris, that means that you cannot expect the immediately adjacent ring of sclera to adapt over time, naturally shrinking to make way for that iris. We would need some other way of increasing the iris's size at the expense of the sclera. The cornea forms the rest of the eyeball's surface. Specifically, it is located at where the light enters the eye, providing an optically clear barrier. It, like the sclera, is mainly made of layers of collagen and other structural molecules. The molecules here, though, are very specifically arranged so that they do not blur or distort incoming light. There are some living cells on the outer and inner surfaces of the cornea, but the vast majority of the cornea's mass comes from non-living structural molecules. As with the sclera, we cannot realistically expect the cornea to remodel and enlarge to correspond with a larger iris. We would probably need to either replace the cornea through a transplant or make some incisions and insert new material around some of its edges to make way for an overall larger cornea. Let's look at where the sclera and cornea meet. Just under where they meet, there are a few tissues with important functions. There's some structures that help drain old intraocular fluid and remove debris, the canal schlemm, the trabecular meshwork, and some tissues involved in the uveoscleral pathway. There's the source for that fluid as well, the ciliary epithelium. There's the ciliary muscles, which anchor and actuate the ligaments that mount and refocus the lens. There's important vascular connectivity, including an artery, the major iridic circle, and several veins. Nerves are present throughout this region. And finally, of course, there's the root of the iris itself, which is to say that we want to preserve the connectivity and functionality of these tissues. So that means don't disrupt this tissue. But that does not mean the tissue wouldn't adapt to shape changes. The tissues that contribute to intraocular fluid management, the ciliary muscles, the blood vessels, and the nerves are all the sorts of tissues that, as we've been learning, actively remodel and respond to changes in their environment. That's a win. Finally, there's the iris itself. The iris surrounds the pupil and can change size to regulate the amount of light coming into the eye. The very posterior surface of the iris is arguably the most important. It's a darkly pigmented membrane that acts as a light stop. Then just anterior to that, there is a layer of smooth muscle that constitutes the dilator muscle. The cells of this muscle collectively form a myoepithelium, and they also have some degree of pigmentation. The dilator muscle's fibers tend to be arranged radially. Around the inner rim of the iris, there is another muscle that opposes the dilator muscle, the sphincter muscle. Here, muscle fibers tend to be arranged circumferentially. Most of the bulk of the iris is made of stroma, many fibroblasts within a loosely packed collagen matrix dominated by collagen type 1. As well, at least for non-blue eyes, there are some melanocytes within the stroma. The most anterior surface of the iris is the anterior border layer. This is actually very similar to the rest of the stroma. It's not a specialized epithelium or anything. There's a layer of fibroblasts and just under them melanocytes that are more plentiful than the rest of the stroma, again, at least for non-blue eyes, but seemingly not any substantially different cell types. There's, of course, some blood vessels and innervation in the stroma, but that about covers it. So for the iris, we've got a living tissue. It's got some non-living matrix components, but still has a meaningful live cell population across all its layers. It's reasonable to expect it to adapt to desired changes in shape over time. We can also expect that changes in gene expression in, say, melanocytes would allow for changes in eye color. The connectivity of the iris is complex, so we wouldn't want to swap out the iris for a new one unless we have to. That said, unlike some other tissues where we've discussed the issues of trying to swap out a highly connected tissue, the iris can work like mostly a closed-loop system, as long as you can engineer a tissue or device to dilate or constrict upon changes in light level. So if changes to existing iris shape are insufficient, it is realistic to consider technologies that aim for an iris tissue transplant or prosthetic. So to review, there's a couple different factors at play: ability of tissue to remodel and whether a tissue can be swapped out or not. The sclera won't remodel, and we're not going to swap it. The cornea won't remodel, but could be swapped. The various tissues, intraocular fluid management, muscles, etc. that surround the lens and iris would remodel, but probably cannot be swapped. And finally, we think that it's reasonable to expect the iris to both remodel and be swappable if needed. So let's figure out how to use these properties. So then, if we need a larger iris, how will we make that happen? It's bounded by the sclera, and as we've learned, we can't just stretch the sclera to widen it. There are three main tissues that would be involved with attaining a larger iris: the sclera, the cornea, and the iris itself. For the sclera, there are three ways we could approach it, leaving more space for a larger apparent iris. The first is to simply implant a ring around the edge, more specifically implanting a ring within or near Schlemm's canal. This is in some ways similar to a type of surgery that is already routinely performed, used as a way of resolving glaucoma. Such a surgery normally applies a ring in tension, pulling inwards. Our ring would instead apply force in the centrifugal direction. This approach would not be expected to change any optical properties. It shouldn't result in the eye's lens being distorted, and overall is technically easy to pull off. However, it would only slightly open up the sclera so that more of the iris is visible. It would be a subtle effect, which may be good enough, but it would be nice to have additional ideas in mind in case larger irises are needed. The second method would be to just build on top of the existing stuff, which is perhaps silly in scientific terms, but it would be technically feasible and provide a lot of flexibility in results. If you imagine layering a new iris-like material on top of the rim formed by the old sclera, and you add some additional new sclera-like tissue off to the sides, you've got yourself a larger appearing iris while leaving all the functional tissues intact. It's worth taking seriously, and might even be the best option. And finally, the third method for dealing with a sclera would be the most aggressive. Recall that the scleral tissue itself is not able to remodel. So let's imagine that you want to get rid of it to make more room for the iris, but you have to keep all the existing connectivity to other tissues, here the blood vessels and nerves, intact. So with this idea, you would use a surgical robot to essentially remove small masses of scleral tissue across hundreds of sites, or perhaps inject specific matrix digesting enzymes, making more room for iris enlargement. It is more speculative, but should be possible in the long term. For a larger cornea, there's two general ways we can approach that. The first would be to swap out the cornea. Cornea transplants are routine surgeries these days, not free of risk, and sometimes the tissue gets rejected since it currently comes from a different donor. So for the sake of a satisfactory and reliable corneal transplant, it could be worth developing a tissue engineering method where patient-derived tissue is grown up in culture and formed into a cornea before being implanted. This field has been advancing, and more recently, even synthetic implants are showing promise as well. The second would be a more limited approach of making an incision around most of the circumference of the cornea, but still leaving the cornea itself centered over the pupil. Then only new engineered corneal tissue or a substitute would need to be provided around the edges. A big benefit there would be that this additional material would not need to have perfect optical properties. And finally, for the iris itself, the actual iris part of this whole iris deal, there's three possible approaches we see. The first, most simply, is allowing the living tissue to gradually expand and remodel as needed. The overall scale of changes is pretty moderate, especially compared to the iris's already impressive range of motion. Fibroblasts, melanocytes, smooth muscle cells, blood vessels, and nerves in the tissue would adapt over time, and new matrix would be deposited as needed over time. The cells will figure it out. The second method corresponds to the silly method for the sclera we mentioned a couple minutes ago. If you build new iris on top of the sclera, you can cheat while leaving the existing physiological functions intact. So here, you would need to provide some new iris-like material to plop on top of the sclera and old iris. The only material you really need to provide is some more fibroblasts and melanocytes and corresponding blood supply, honestly. No need for innervation since the old iris's musculature will still be there. This would be a passive tissue just there for appearances. Or you could even design something like a soft silicone substitute. The final method would be to swap out the iris tissue for a new iris. The iris is a fairly self-contained tissue, and as long as you can get a new tissue to expand and contract in response to changes in light level, that's all you really need out of the engineering process. And you can imagine ways of doing that that don't even depend on natural innervation. As you can see, plenty of options exist for getting the sclera, cornea, and iris itself to cooperate for accomplishing the appearance of a larger iris. If a larger iris turns out to be beneficial for an anthro's appearance, it can be done in a satisfactory way. Aside from general questions of changing the size or shape of irises, we can imagine there are a couple ways that people might want modified pupils. First, they might want different pupil shapes, and second, they might want somewhat larger appearing pupils. For changing pupil shape, all three of the iris modifications we just mentioned would be applicable. Gradually forcing the iris to change its shape as it remodels, layering new iris tissue on top, or swapping the iris for a new engineered one. Specific engineering methods are beyond the scope of this video, so we will pause there for pupil shape modifications. For pupil size, as you know, pupils dilate or contract according to light levels. In addition to all the various remodeling and tissue engineering approaches we've discussed, there is another neat way this could be approached. To get larger pupils, if you want someone to appear to have larger pupils, you can figuratively have them wear sunglasses of low to moderate strength. To be a bit more proper, you can apply a neutral density filter just under the cornea, slightly reducing the overall amount of light entering the eye at any given outside light level. The pupils will increase in diameter to compensate. This will force an increase in the pupil's aperture size and will have a modest effect in reducing the depth of field for the photographers out there. But based on an in-house proof of concept experiment, we suspect that a modest increase in pupil diameter can be attained without perceptibly affecting someone's vision. We've saved the easiest for last. If you want to change iris color, given that there are living melanocytes in the iris, all that you theoretically have to do is apply a genetic treatment to those melanocytes to change the amount or type of melanin they are expressing. Or perhaps you can derive some additional melanocytes from patient stem cells, work with them in vitro, and implant them. If you want to go from a pigmented iris to something that is lighter in color, it might require selective ablation of melanocytes, which would involve a different kind of genetic treatment. As we have seen, tissues in the eye vary in how much they can remodel, as well as the degree to which we can safely replace them if needed. The sclera will not allow for remodeling, nor can we replace it. The cornea can't be remodeled, but has good prospects at being swapped out or supplemented with additional transparent material if needed. Other nearby tissues that anchor the iris and that maintain intraocular fluids can be counted on to reshape if needed, though their connectivity should not be interrupted. And the iris itself is expected to at least have some remodeling capacity, as well as some amenability to be replaced with an implant or prosthetic. Working within these considerations, we were able to propose several ways of attaining a larger appearing iris, either at the expense of the sclera or by building on top of the sclera. As well, we discussed ways that pupil shape and size can be modified, and briefly mentioned how the iris can be targeted for various ways of changing eye color. Again, based on the anatomical model we showed in the first video in the series, it appears that the iris might not have to be larger, at least in wolves. Still, it may be beneficial to have a capability for larger irises, especially for other species. It would also be useful to have options for modifying the pupil as well as changing iris color. Overall, the prospects of changing the eye's appearance are very promising. We're almost done. As we mentioned when starting this video, we don't have the space to discuss a few other types of tissues. We can't cover adipose tissue, the mucous membranes of the nose and throat, or secretory glands such as the salivary glands. While we did mention in Chapter 1 that things like the nasal passages would need to be larger, like for a longer snout, these tissues tend to have simple or repetitive geometry, have shapes that fit into whatever spaces are provided for them, are surgically accessible, and have a good degree of self-repairing ability. Additionally, we're not going to discuss the voice box here. We can't exclude the possibility that a change to the trachea's outer shape, whether additive or subtractive, like a tracheal shave, might be helpful for the overall shape of the head and neck. And we will also point out that until some detailed modeling is performed of how vocalization will change with a longer snout, pitch modifying surgeries to the voice box may or may not be relevant. We don't know yet. But in all cases, these types of surgeries, if they do wind up being needed, are pretty well developed in current practice, so we don't need to discuss them in this video. Time to put everything together. In Chapter 1, we analyzed the large-scale anatomy and physiology of an anthro's head. A satisfactory result in appearance and in function can be achieved while staying within constraints imposed by things like the brain case and placement of the inner ear. Then the big question was, how do you get to that anatomy? Across chapters 2 and 3, we have analyzed the types of tissues that need change for an anthropomorphic transition procedure. Building off the analysis of chapter 1, we broke down the anatomy into smaller parts, looked into the relevant biology of the tissues, and made proposals for how the changes could work. Between Chapter 2's video and the video that you're watching now, the most important conclusions from this analysis are, first, in terms of bones and related tissues, bones will be compatible with all sorts of manipulation. In contrast, entheses and ligaments aren't going to be as nice. Luckily, they generally do not need to be changed, and we were able to make some proposals for a few cases where necessary changes came up. Next, for cartilage, new implants will be needed for the ears and nose, but these are expected to be relatively easy for tissue engineering. Moving along to muscles and tendons, we expect that existing muscles will generally automatically adapt as needed. Some tendons might need special handling, but they can be manipulated in a way that preserves their mechanical strength. And for the ears, new muscles and tendons will be needed, but they face relatively low mechanical demands and would be appropriate to control by electrodes. Then for the nervous system, as a general rule, we want to keep existing neural connections to sensory and motor organs in place. We grappled with the fact that a few critical nerves will need to be lengthened. Thankfully, axon extension can be relied upon up to limits imposed by the distances between nodes of Ranvier and myelinated axons. And finally, for the eyes, developing capabilities for larger or differently shaped irises and pupils would be beneficial even if wolves might be able to get away without it. There are a few directions that could be pursued for these capabilities, even though we can't count on the sclera or cornea to remodel automatically. And in terms of changes to iris color, there are a few techniques that can be considered, including targeting the melanocytes in that tissue. Of course, you can imagine there's a lot more information you would need before you can develop and use the necessary technologies like tissue engineering and surgical techniques. The goal with these videos was to introduce relevant technologies to at least get that ball rolling. And we mean that seriously. Considering the diversity of technical challenges here, it is important to provide a shared base of knowledge and some targets to work towards to folks who are actually working on this stuff. This is absolutely doable, and you can get a great result as long as you break things down into smaller parts. Okay, folks, that will do it. Wow. Thank you so much for sticking with us as we took this tour de force through some really intense details. We hope that you enjoyed this and found it fascinating. These videos were beasts to write and produce. It was equally challenging and rewarding and challenging to put all this information together and to make the best, most informed decisions we could. Thank you in particular to Medic, who helped with some of the analysis for the iris and cornea, and who did a technical review for the rest of the script. And thank you, of course, to Lathrius again for making the 3D wolf model possible in the first place. Thank you more broadly to folks in our community for many helpful and constructive comments, and to supporters on Patreon. If you would like to join our community, you can go to our website and follow the links from there. And if you would like to support our work, please consider joining as a member on Patreon. Thank you so much for your consideration. And P.S. While this video series has reached its conclusion, we'll be doing more in the future, so be sure to subscribe if you don't want to miss it. Thanks again for watching. Catch you all later.